Okay, actions of antibodies. Okay, how the antibodies destroy the antigens. So antigens are destroyed by antibodies through several mechanisms illustrated in these figures 11.6. Okay, so the first one we have agglutination. So you have to imagine lah. You use your imagination to imagine lah what happened. Okay, so agglutination. So antibodies coagulate the pathogens and make them an easy target to be trapped and destroyed by phagocytes. So this antibody will bind. Okay, will bind together the antigen and make it become make it and coagulate them. So for example, so I have to draw lah. Okay, let's say this is our blood vessel, for example. Okay, so the antigen enter. Okay, antigen enter like this. Okay, so this one is the phagocyte. Okay, so if the antigen enter like this, so phagocyte have to go one by one. Okay, it has to destroy this one, then it have to go to this one, destroy, go this one, destroy. So actually we taking longer time lah. So what happened? Okay, this agglutination, so I draw back. Okay, so agglutination, so this is the phagocyte. So these antibodies make these pathogens coagulate together, so they will bind these pathogen to each other, so become coagulate. So this is really for this phagocyte to trap. So it can trap maybe like this. Okay, can trap the pathogen and destroy the pathogen easily. So this is the function of antibodies. So antibodies help to coagulate the pathogens to make them clot like this. So they're easy to be trapped and destroyed by phagocytes. So this is for the first one, agglutination. Okay, the action of antibody. So sometimes the antibody not really destroy the antigens. Actually, it will help the phagocyte. Okay, help the phagocyte to destroy the pathogens. Okay, this is for the first one. Okay, now we have the neutralization. Okay, neutralization. Yeah. Wait, neutralization. Okay, for neutralization, okay, I also will draw here. Happen. It cannot. Wait, Okay. Okay, for this neutralization, okay, antibodies will bind with toxin produced by bacteria and neutralize the toxin. So let's say, okay, we draw this one in our blood vessels. Okay, our blood vessel. So this is the bacterium. Okay, for example, this is a bacterium of pathogens. Okay, we produce, okay, release, okay, or produced by this release the toxin. So it will release toxin lah. Toxin like this, right? See this. So this is the toxin. Okay, toxin produced by the bacterium. So what happened? The antibody here will go to this go to this toxin like this, and then it become like this lah. Will bind with toxin and then neutralize it. This one not help the phagocyte lah, but this one actually directly this antibody will neutralize the toxin. So make this toxin not become harmful lah. So this is the directly, but the toxin actually released by the bacterium or by the pathogens. So the antibody will react with the toxin and then neutralize it. So this one not involve phagocyte. Okay. Okay, so this one neutralization, and then we have this one is the precipitation. Okay, precipitation. For example, okay, example. Okay, let's say we have this our blood vessel. So the antigen enter, but the antigen cannot be seen or cannot be recognized by phagocyte because it's been dissolved. Okay, dissolved antigens. So when something dissolve, we cannot see, right? For example, if you dissolve the sugar inside the water, you cannot see the sugar. You just can only see the water, right? When the sugar completely dissolve in the water. So this also same. So dissolved antigen cannot see or cannot recognized by the pathogens. So this may be this by the phagocytes, not pathogens. Okay, so this may be the phagocyte. Maybe the phagocyte not even know there is antigen there. So what happened? This is the action of antibody. So the antibody here will then go to these antigens. Okay, I'll just write the antibody. Okay, antibody. Okay, go to this antigen. So this is what happened. Okay, it will make this react with dissolved antigen to form insoluble complex. So make this dissolved antigens become insoluble complex or become precipitate. Become precipitate means like this lah. Maybe now the become a particles like this. Like you not dissolve lah. You just put the sugar inside the water so you can see the sugar, right? But you not dissolve it. So that's same lah. So you can see the sugar. So this one you can see the this pathogen phagocyte now can recognize these antigens and then it can destroy these pathogens. Compared to this structure, dissolve this phagocyte cannot recognize. But after the antibody reacts and make it become insoluble complex or precipitate like this, so now this phagocyte can recognize and then can track them and then can destroy. Okay, this is precipitation. Okay, dissolve. The word here from dissolve to insoluble complex. Precipitate. Okay. So that's for the third one. And the fourth one, we have this opsonization. What is opsonization? Antibodies combine with antigens and act as a marker for phagocyte to recognize the antigens and destroy them. Okay. Say this is our blood vessel. So bacterium enter with the antigen on the surface. So this phagocyte cannot recognize. Okay, cannot recognize the antigen. So what happened? The antibody then will bind. Okay, combine with the antigen here, and then act as a marker. So when the antibody combine with this antigen, so now the phagocyte can recognize the antigen. So opsonin, the name of antibodies, opsonin antibodies combine with antigens. So now this phagocyte can recognize. So now it can destroy them. If like this phagocyte cannot recognize. But when the antibody binds with the antigen, this phagocyte can recognize. So it can destroy the bacterium. Tried the bacterium that has been identified by the antibody marker. So this opsonization. Like the last one is the lysis. Antibodies combine with antigens and cause bacteria to be broken down and decompose. So, lysin antibody combines with antigen. Okay. Then this make bacterium undergoes lysis, decomposition. So before this, maybe the bacteria okay like this, right? With the with the antigen. Okay. But when it combine, this lysin antibody combine with this antigen, so it make the bacteria broken down. So this is known as lysis.